In this video, I want to talk about the GPCR G-alpha-S adenylate cyclase cyclic AMP protein kinase A signaling pathway. This is one of the most common G protein coupled receptor pathways. It's activated by a number of neurotransmitters and hormones and plays some important roles in physiology. Let's go ahead and look at the pathway. Well, this is a GPCR coupled pathway, which means that the receptors that activate it will be GPCRs, G protein coupled receptors, members of that large family, all of which have those 7 membrane spanning domains and which interact with the heterotrimeric G protein class. So here's our GPCR in the membrane, and here's the heterotrimeric G protein with the alpha, the beta, and the gamma subunits. And for this particular signaling pathway, we're going to be looking at the G-alpha-S subclass. Well, what happens when we add the signal? Signal here is represented by the purple dot. It's going to come, bind to the receptor, and when it does that, it will induce a conformational change. Because the GPCR is closely associated with the G protein, when the GPCR binds its ligand and changes shape, it induces a change in the GPCR as well. And specifically what's going to happen is the alpha subunit is going to release the GDP and instead pick up a GTP. Once the alpha subunit is bound to a GTP, it loses the affinity for the beta and the gamma subunits, and because the gamma subunit is helping it associate with the GPCR, it will also dissociate from the receptor, and the beta and the gamma subunit will diffuse away through the membrane, and the alpha subunit, still bound to the GTP, will also diffuse away through the membrane. Let's have a look at what happens next. I'll leave the beta and the gamma subunits for another video, and in this one we'll look at what happens with the alpha subunit. The main target for the G-alpha subunit is an enzyme called adenylate cyclase. It's also called adenylyl cyclase, commonly abbreviated AC. This is a membrane protein, and the reaction that it catalyzes is to take ATP as a substrate and then convert it into another compound called cyclic AMP. So let's take a quick look at ATP. Here I've drawn it in cartoon form, and I just want to point out that ATP is a nucleotide. It has an adenine, that's the nitrogen-containing base, ribose, and then these 3 phosphate groups. The reaction catalyzed by adenylate cyclase converts ATP into a molecule of cyclic AMP and also releases 2 inorganic phosphates. Cyclic AMP is shown here and in cartoon form as well, and I want you to notice that cyclic AMP is also a nucleotide. It has adenine, it has the ribose, it has only 1 phosphate group, and that phosphate is bound to the ribose by 2 covalent bonds, and that forms sort of a ring structure. This is why we call it cyclic AMP. Alright, let's return and see what happens in the cell. Well, the activated G-alpha subunit diffuses through the membrane. It will interact with the adenylate cyclase molecule, activating it. And once it's activated, what will happen is the cell will start to take ATP and convert it into the product, cyclic AMP. And enzymes can catalyze many reactions per second, so a lot of cyclic AMP will be produced. Now, cells normally have ATP present, but they do not normally have cyclic AMP. That's an unusual compound, and so when it shows up, it's a sign that something has happened outside the cell. What does the increase in cyclic AMP do in the cell? Well, cyclic AMP is a small molecule. It can dissolve away and it can interact with a number of different targets, but the primary target in this signaling pathway is an enzyme called protein kinase A. In the inactive form, protein kinase A is a tetramer. There are 2 regulatory subunits and 2 catalytic subunits, and it's off. And what happens when there's an increase in cyclic AMP is 4 cyclic AMP molecules bind to the regulatory subunits, 2 to each one of them, and that causes the regulatory subunits to dissociate from the catalytic subunits. We get the release of 2 active PKA molecules, and those kinases then can phosphorylate target proteins. Protein kinase A phosphorylates a number of different proteins in the cell. There are some that are in the cytoplasm, there are some that are in the membrane. In addition to those targets, protein kinase A can move into the nucleus, and there's a particular protein there called CREB, which can be activated by phosphorylation. CREB is a transcription factor. It's normally inactive, but if it gets phosphorylated by the activated protein kinase A, then CREB can sit down on specific DNA sequences and turn on or off gene transcription. And what that means is that there can be long-lasting effects because we can turn on a whole new suite of proteins in response to an external signal. I want to say a word about pathway and activation. Cells need to have a way to turn pathways off, and there are a number of ways in which this particular pathway can be shut off. Maybe the simplest thing is that if the ligand is removed, if that purple signal is removed, then what will happen is the GPCR will be returned to its inactive configuration. Another thing that happens is that the alpha subunit of the G protein has an intrinsic low GTPase activity, and what that means is it will take the GTP and hydrolyze it to convert it into GDP. And when that happens, it will lose affinity for the adenylate cyclase. It will diffuse through the membrane. It can reassociate with the beta and the gamma subunits and ultimately return to the GPCR to await a new signal. So this would be hydrolysis of the GTP in order to inactivate it. A third mechanism is to break down the cyclic AMP. Cyclic AMP can be converted into a compound called AMP by the enzyme phosphodiesterase. It breaks one of those covalent bonds linking the phosphate group to the ribose. So the molecule is similar, but now it's no longer cyclic. It's just plain old AMP. And regular old AMP cannot interact with the regulatory subunits of protein kinase A, so that molecule will be returned to its inactive state, and this shuts off the pathway. There's one last mechanism to talk about, and this is something called desensitization. It can happen when there's prolonged stimulation, and it's going to involve a molecule called Gerk, G protein coupled receptor kinase. All right, well, why is this necessary? Well, let's suppose that the cell is seeing a chronically high level of ligand. Maybe the purple signal is always there. Well, this can pose a problem over time, and here's why. Earlier I said that the alpha subunit can hydrolyze GTP in order to turn it into GDP. But if the signal is always around, what will happen is as soon as it recombines with the activated GPCR, the one that has that ligand bound to it, then what will happen is the alpha subunit will exchange the GDP for a GTP, and the whole pathway would start again. So it can be advantageous for the cell to shut this off so that we don't have that repeated stimulation and then activation. This is where Gerk comes in. So Gerk, that G protein coupled receptor kinase, is normally off, but it can be phosphorylated, and when it is, it becomes activated. And one of the targets for Gerk is the GPCR. It can go and phosphorylate the G protein coupled receptor on the cytoplasmic side. When this happens, it allows another protein called arrestin to come and bind there. And binding arrestin will prevent the heterotrimeric G protein from being able to come in and interact with the GPCR. How does Gerk get phosphorylated? Well, one of the enzymes that can do this is protein kinase A. So once this pathway is turned on, there can be a feedback loop that will lead to it being turned off. In addition to blocking the interaction of the GPCR with the G proteins, arrestin can serve a number of different functions. One of the things that happens is when arrestin binds to the GPCR, that receptor arrestin complex gets brought into the cell through receptor-mediated endocytosis. What happens to the receptor and the arrestin varies. Sometimes what happens is it's returned to the membrane, so the receptor can be recycled. We call this resensitization when it gets returned to the membrane. However, other times what happens is the receptor fuses with a lysosome and the receptor protein is broken down completely so that the receptor numbers will fall. This mechanism is important because it can lead to fewer receptor molecules being present on the cell following application of a drug. It might be one of the reasons why we see in pharmacology that some medications stop working after a while because the cell has responded by downregulating the receptors that the drug would target. There's a third mechanism that arrestin's involved in, and that's that the arrestin GPCR complex can initiate another wave of cell signaling. This can happen because the arrestin GPCR complex can recruit proteins. For example, it might recruit proteins and activate kinases. It can act as a scaffolding system so that other proteins can be recruited to this area. For example, arrestin GPCRs can lead to changes in the cytoskeletal structure. It can even activate other cell signaling pathways, and these changes can take place at the cell surface, but even after the GPCR arrestin complex has been brought into the cell in an endosome. What I want you to take away from this is how a single signal outside the cell can lead to a whole host of changes in the cytoplasm of the cell.